All right, welcome to The Explainer. Today we are diving deep into one of the biggest scientific stories ever told. We're talking about the hunt for the blueprint of life itself, the story of DNA.
So here's the game plan. We're going to start with the big mystery, right? What molecule actually carries our traits? From there, we'll jump into the absolutely thrilling race to figure out its shape, then decode how it's read, and finally, see how this one discovery just completely changed everything.
Okay, so our story kicks off not with some big revelation, but with a massive question. A question that had scientists totally stumped for like the better part of a century.
What is life's instruction manual made of? What molecule could possibly hold all the information needed to build a living, breathing thing? For the longest time, honestly, nobody had a clue. But as we got into the early 20th century, science finally started piecing together enough clues to get a real debate going.
And you can see how science was really closing in on it. We knew traits got passed down through these factors, as Mendel called them. And thanks to all those fruit flies, we knew those factors, we call them genes now, were located on our chromosomes. But here's the catch. Chromosomes are made of both protein and DNA. And this kicked off a huge scientific argument.
And you've got to appreciate the logic they were using back then. Honestly, the smart money was all on protein. It's made of 20 different complex amino acids. It just seemed like it had to be the one, complex enough to store all the secrets of life. DNA, on the other hand, with its super simple four-letter alphabet, it just looked, it looked too simple for such a massive job.
But then, boom. In 1944, after a quiet decade-long research project at the Rockefeller Institute, a bombshell dropped. Oswald Avery and his team, Colin MacLeod and Macklin McCarty, they came forward with evidence that was just incredibly difficult to argue against.
And their experiment was just so clever. They took harmless bacteria and transformed them into a deadly version, just by using an extract from some killed lethal bacteria. Then, they basically started a process of elimination. Destroy the proteins, the transformation still happened. Destroy the RNA, yep, still worked. But the second they added an enzyme that destroyed the DNA, the transformation stopped, dead in its tracks. That was it. That was the smoking gun. DNA was the molecule.
So that was it. The debate was over. Science had its molecule. But, knowing what the blueprint was made of didn't mean they could actually read it. To do that, they needed to know its shape. And that, my friends, set off the next great scientific race.
Yeah, this question, if DNA is the blueprint, what does it actually look like, kicked off a totally fierce international race. And solving this thing, it wasn't the work of just one lab. It was all about fitting together these crucial puzzle pieces from all over the world. Okay, so the first major piece of the puzzle came from a biochemist named Erwin Chargaff. He started analyzing DNA from all different species and found this really strange but totally consistent pattern. Just look at the numbers here for humans and then for E. coli. The amount of adenine, or A, is always, almost identical to thymine, T, and guanine, G, always matches up with cytosine, C. This wasn't some fluke, it was a fundamental rule of life.
And the final, absolutely decisive clue? That came out of King's College London. A scientist named Rosalind Franklin, using this technique called X-ray diffraction, which is basically a way of taking photos at a molecular level, she produced this incredible image. We now know it as Photo 51. And its pattern was just unmistakable. That clear X shape, it practically screamed helix. Her data also hinted that it wasn't just one strand, but probably two.
So, with Chargaff's rules in one hand and the incredible insights from Franklin's data in the other, two researchers over at Cambridge University, James Watson and Francis Crick, put it all together. And in 1953, they proposed their now iconic model, the DNA double helix.
The absolute genius of this model was that the structure itself was the explanation. It just made perfect sense. This complementary pairing, A always with T, G always with C, meant the two strands were like perfect mirror images of each other. And right away, that suggested how DNA could copy itself. All you have to do is unzip the strands, and each side becomes a perfect template for a new one. This beautiful, elegant process, we call it semi-conservative replication.
Okay, so we have the blueprint, we know what it looks like. But how does a static code just sitting there on a molecule actually do anything? How does it become the thing that directs all of life's activities?
Well, the flow of all that genetic information is governed by something we call the central dogma of molecular biology. It's a two-step process. First, the main DNA code is transcribed, or copied, into a temporary message. That's messenger RNA, or mRNA. Then, that message is translated into a protein. Think of it like a two-step courier system.
So that first step, transcription, that's handled by a little molecular machine called RNA polymerase. It's amazing. It latches onto the DNA, unwinds a small section of it, and then synthesizes a matching strand of RNA. It's basically making a photocopy of the gene's instructions.
Then that mRNA message, that little photocopy, it travels out of the nucleus and into the main part of the cell, where it finds a ribosome. Now, you can think of the ribosome as the factory floor. It's the place where the final product, the protein, is actually going to get built. Now, here's where it gets really cool. The ribosome doesn't read the message one letter at a time. It reads it in three-letter words that we call codons. Each of these unique three-letter words specifies one of the 20 different amino acids. And for a bit of trivia, the very first codon they cracked back in 1961 was UUU, which they figured out codes for the amino acid phenylalanine.
You know, this fundamental understanding, how genes become proteins, it did way more than just solve a puzzle. It literally gave humanity a new instruction manual for life. And it launched the absolutely revolutionary field of genomics.
And by 2003, they had done it. Scientists had sequenced the entire human genome. You see that number on the screen? 3 billion. That's how many letters are in our genetic code. And let me tell you, having the full script in our hands led to some seriously surprising revelations.
Yeah, one of the biggest shockers was this. Having more DNA does not mean you're more complex. It's all about something called gene density. Just look at this. Bacteria, they cram almost 900 genes into a million base pairs. A fruit fly, around 100. And humans, we have a measly 7. Turns out most of our DNA isn't even coding for proteins. It's acting more like a super complex regulatory switchboard, telling genes when to turn on and off. And this, right here, is why cracking the code matters so much. This isn't just for textbooks. It has completely transformed our world. We can now pinpoint a single letter change in our DNA that causes a disease, like sickle cell anemia. We can compare our genomes to other species to trace our evolutionary past. And, incredibly, we can design powerful new medicines that go straight to the genetic source of an illness.
So this whole incredible journey, from a simple garden patch with Mendel's peas all the way to a complete map of our genome, it's given us this unbelievable power. The power to not just read, but even rewrite the very language of life. And that leaves us with maybe the most important question of our time.